Have you ever cut your skin and it healed? If so, you have cell division to thank. When you cut your skin, some of the cells are removed. Surrounding skin cells receive signals to start dividing to fill in the missing cells. Your body is constantly undergoing cell division to replace the 50 to 70 billion of your worn out or damaged cells that die per day, in addition to another 2 million red blood cells that die per second. This equates to a total of 222 to 242 billion cells per day that must be replaced by cell division. Cell division is the process by which one cell gives rise to two or more cells. We start with a parent cell that divides to yield daughter cells. Cell division is essential for reproduction, growth, and repair of all organisms. There are three types of cell division that take place in different types of organisms. The first is binary fission that occurs in prokaryotes. Prokaryotes are single-celled organisms, including bacteria and archaea. During this process, one parent cell divides into two daughter cells that have the same DNA. The same DNA is found in both daughter cells as well as the original parental cell. Mitosis takes place in both unicellular and multicellular eukaryotes. Remember that eukaryotes contain specialized organelles or structures like a nucleus and endoplasmic reticulum. Mitosis is used to generate non-gamete cells. In this case, one parental cell also divides to yield two daughter cells that each have the same DNA. Meiosis is a specialized type of cell division that takes place in multicellular eukaryotes only to yield gametes, which are egg and sperm cells. In this case, one parent cell divides to yield two intermediate cells that then divide to yield four daughter cells that have half the amount of DNA as the original parental cell. Single-celled organisms utilize cell division as their means of reproduction to generate offspring. This is known as asexual reproduction, during which one cell copies its DNA and divides into two genetically identical offspring cells that each receive one copy of the DNA. Prokaryotes reproduce via binary fission, whereas unicellular eukaryotes like yeast reproduce via mitosis. Multicellular eukaryotes like plants and animals undergo sexual reproduction. In this case, male and female gametes from two parents fuse to produce genetically different offspring. Prokaryotes, including bacteria and archaea, are single-celled organisms that divide via binary fission. If you look at the figure on the left, you see a typical bacteria cell with one chromosome in the nucleoid region. On the right, what we see is that this single chromosome in the nucleoid region duplicates or copies, and then the cell splits in half to yield two daughter cells, each with one of the chromosomes. Gametes are generated by meiosis. Remember that gametes are egg and sperm cells that contain half the amount of DNA as their parent cell and the rest of the non-gamete cells in an organism. If you are female, your gametes are your egg cells in your ovaries. If you're male, your gametes are your sperm cells in your testes. The rest of the cells in your body are non-gametes. The purpose of gametes is fertilization, in which a sperm fuses with an egg cell, and the DNA from both cells merge to yield a zygote that contains a full complement of DNA. So the offspring has half of its mom's DNA and half of its dad's DNA. So half from mom plus half from dad equals 1, or a full complement of DNA. Imagine if the gametes contain the full amount of DNA. If gametes contain the full amount of DNA, this means a zygote would receive twice the number of chromosomes that it needs. Once a zygote is generated, it undergoes mitosis and cellular differentiation to develop into a baby. All multicellular organisms, including the sea urchin, begins life as a single cell that divides to form two cells. After 4 rounds of cell division, there are 16 cells. After many rounds of cell division, the individual develops into a complex multicellular organism seen in this mature sea urchin. Mitosis serves many purposes in eukaryotic organisms. During this process, one parent cell divides to yield two daughter cells that have the same DNA. This process is used to generate non-gamete cells. In multicellular organisms, mitosis of a zygote and embryo produces a baby organism. After the baby is born, cells continue to undergo mitosis for a baby to develop into an adult. Keep in mind that human adults have somewhere between 10 to 100 trillion cells. Therefore, we all start as the fusion of a sperm and egg cell, so in other words as one cell, and by the time we reach adulthood, our cells have divided enough to yield 10 to 100 trillion cells. Even throughout adulthood, our cells continue to divide. For example, in our skin, cells flake off constantly, and it's estimated that your entire skin is replaced every single month. When you cut your skin cells get removed that also get replaced via mitosis. Many rounds of mitosis take place to allow a baby to grow into a toddler, into a child, and into a young adult. The larger the human is, the more cells that they have. The outer layer of skin cells is always flaking off and being replaced with newer cells dividing beneath the surface. The life cycle of multicellular organisms includes both meiotic and mitotic cell division. In an adult, meiosis generates gametes that fuse during the process of fertilization to yield a zygote. The zygote then undergoes mitosis to generate a baby. The baby undergoes mitosis to grow into an adult, where the process starts over. The adult now undergoes meiosis to start producing new gametes, and so on and so forth. In humans, adults form egg and sperm cells in specialized organs, the ovaries in females and the testes in males. These gametes contain half the number of chromosomes, so they are called haploid. Fusion of these gametes during fertilization yields a zygote that contains both sets of chromosomes from mom and dad, which means that they are diploid, referring to the fact that they contain two sets of chromosomes. The zygote then undergoes multiple rounds of mitosis to produce a multicellular offspring and eventually an adult where the cycle starts over. As we discuss cell division, we also need to realize that cell death is a normal part of development and repair in an organism. This process of programmed cell death is referred to as apoptosis. This is important in embryological development and for removing damaged cells. For instance, during embryological development of birds, all birds have webbing in between their toes. This webbing remains in water-dwelling creatures such as ducks, but gets removed via apoptosis in land-dwelling birds like chickens. What about humans who get a sunburn? This means that the outer layer of skin cells is damaged, so they undergo programmed cell death and peel away. Human cells are genetically programmed to divide about 50 times and then die. This prevents cells from accumulating too many mutations and continuing to divide and remain in the body. Alterations in this program can lead to cancer, which is discussed more in Part B of this lecture. Sunburnt skin dies and peels off, being replaced by healthy new cells underneath the surface of the skin. During mitosis, a parental cell divides to yield two daughter cells that have the same DNA. Keep in mind that cells need a complete set of genetic information known as the genome to survive. Therefore, a cell that receives less than a full set of DNA will not be able to survive. When a cell divides, all cellular components, including a complete set of DNA, must be passed to the two daughter cells. Therefore, before division, a cell copies its DNA so that it has two complete sets, one to pass down to each daughter cell. So once again, overall, during mitosis, one cell divides to yield two daughter cells with the same DNA. During the process of DNA replication, two identical double helix molecules are formed prior to cell division. Keep in mind that DNA is a double helix molecule, so when it copies itself, it yields two double helix molecules. Replication results in one parental strand pairing with one brand newly synthesized daughter strand. This is referred to as semiconservative replication because the daughter DNA molecule conserves one parental strand. So semi means partial and conserve means, well, to retain. So the daughter molecule retains one of the parental strands or part of the parental molecule. A copy of each daughter molecule gets passed to each daughter cell. Watch the following video to learn more about semiconservative replication. DNA replication produces two identical double helix molecules. Each parental strand in blue is used as a template to build complementary daughter strands shown in red. In looking at the two daughter molecules, you can see that they are identical. You can start at the top and read each base pair moving downward to convince yourself. During DNA replication, one molecule is copied to yield two. The gray parental strands separate and are used as templates to synthesize new daughter strands. The resulting molecules have one parental strand in gray and one newly synthesized strand in blue, which is why this is called semiconservative replication. Several enzymes catalyze the steps of DNA replication, and these enzymes are numbered on the next few slides. DNA helicase breaks the hydrogen bonds between the bases to pull the two DNA strands apart and to unwind the double helix. Replication bubbles are formed at various points along the chromosome where localized replication takes place. By forming multiple replication bubbles, this allows replication of a long chromosome to take place much more quickly. Where each replication bubble is formed and replication begins is referred to as an origin of replication, since origin means to begin. Replication forks are found at the end of each replication bubble where the parental strands have not yet unwound. Origins of replication are found along the length of the chromosome that open into replication bubbles where localized replication takes place. Eventually, the replication bubbles meet to give continuous strands of DNA. Once DNA helicase pulls apart the two DNA strands, DNA polymerase can gain access to the parental strands to synthesize new DNA strands that are complementary to the original parental strands. DNA polymerase adds nucleotides with complementary bases to each exposed parental strand, matching adenine with thymine and guanine with cytosine. DNA polymerase also links the phosphate of the incoming nucleotide to the sugar of the previous nucleotide. Since different segments of the chromosome are replicated at each replication bubble, DNA ligase comes along next to link the segments together to give long continuous strands. DNA helicase breaks the hydrogen bonds between the bases to pull the two DNA strands apart and unwind the double helix. DNA polymerase synthesizes DNA strands complementary to the parental strands. DNA ligase links discontinuous segments together to give long continuous strands. DNA polymerase matches incorrect base pairs once every 10,000 base pairs. This is due to the speed of replication, as polymerase adds up to 50 nucleotides per second. Therefore, DNA repair enzymes proofread each daughter strand after replication to repair the errors. The completed DNA strands contain only one mistake every billion base pairs after proofreading. Given the fact that human cells have 3 billion base pairs total, this means on average, after DNA replication takes place in each one of our cells, about 3 mistakes remain in our DNA. DNA polymerase has proofreading activity that corrects errors during replication. However, many errors are missed, therefore DNA repair enzymes conduct further proofreading after DNA polymerase to correct many of the originally missed mistakes. In mismatch repair, depicted in the upper right-hand corner of the figure, the incorrectly added base is detected after replication. The mismatch repair enzymes remove the incorrect nucleotide from the newly synthesized strand and replace it with the correct nucleotide having the correct complementary base. Mistakes during replication that are not repaired cause mutations, which are changes in DNA sequence. Although it sounds like mutations are a bad thing, they're not always bad. They actually lead to genetic diversity that promotes survival. For instance, if a disease hits the human population, genetic diversity should allow at least a certain percentage of the population to survive. Also recall that environmental factors, including ultraviolet light from the sun or chemicals in cigarette smoke, can also damage DNA and cause mutations. If we take a look at the genetic information in your cells that must be copied prior to cell division, we can talk about DNA, genes, and chromosomes. DNA is deoxyribonucleic acid. It contains the hereditary information and is composed of nucleotides that are strung together to give a longer molecule. Recall that DNA has a double helix shape. Double refers to 2 strands of nucleotides, and helix refers to being coiled like a corkscrew or a slinky. Genes are particular sequences of DNA on chromosomes that usually encode proteins. Chromosomes are long DNA molecules that contain hundreds to thousands of genes. DNA has a double helix structure that contains nucleotides with one of 4 bases: thymine, adenine, guanine, and cytosine. Each species has a characteristic number of chromosomes. Bacteria, which are single-celled organisms, only have 1 chromosome. If we compare this to humans, humans have 46 chromosomes in each non-gamete cell and 23 chromosomes in each gamete. Recall that the gametes are egg and sperm cells and contain half the number of chromosomes as all of the non-gamete cells in your body. If you were to take the chromosomes out of one of your cells and stretch them out end to end, they would be about 6 feet long. Therefore, you have 6 feet worth of DNA inside each one of your microscopic cells. For this reason, proteins bind to the chromosomal DNA to coil and condense the strands so they fit inside your cells. This DNA protein complex is referred to as chromatin. The proteins in particular are called histone proteins. Nucleosome refers to a section of DNA wrapped around these histone proteins. During cell division, the chromosomes condense even further to make them easier to move, which we will discuss in Part B of this lecture. Double-stranded DNA wraps around histone proteins to form nucleosomes that have the appearance of beads on a string. The nucleosomes are coiled into a chromatin fiber. When a cell undergoes mitosis, the chromosomes condense even further. Remember that prior to cell division, the DNA in a cell must be copied. The DNA is in the form of chromosomes, which are long stretches of DNA with different genes. So when DNA is replicated before cell division, this results in what's called a duplicated chromosome. A duplicated chromosome has 2 DNA double helices attached at what's called a centromere region. Each DNA molecule within a duplicated chromosome is called a sister chromatid. A duplicated chromosome has 2 identical sister chromatids that are held together at the centromere region. Mitotic spindle microtubules attach to a disc called the kinetochore. Spindle microtubules will move the chromosome, which we will discuss more later in Part B of this lecture.